Hello, everybody, and welcome to my science fair project. My name is Aderemi, and today I'll be presenting Regeneration. The contents of this slideshow include the problem, methodology, research, hypothesis, and right before my conclusion, the data. I will also have a citations and acknowledgement slide. My problem definition is that I wonder if maybe we can use mitosis or genome editing under laws and regulations to help those who have experienced limb amputation. Mitosis is a cell division process, but uncontrolled mitosis can lead to cancer, but it may be the key to regrowing a limb. Genome editing is another possibility. It is banned in most countries because gene editing on healthy embryos may lead to irreversible mutations of the future hereditary of that same embryo. But we may need it for regeneration. So my scientific question is, can scientists cure limb disabilities in the future using mitosis and or genome editing? My methodology will be following the steps below: the causes of regeneration, mitosis in mammals, mitosis in reptiles, intra and post embryonic stage events for mammals and reptiles. I will also study if there are any genes required to take out or leave alone, or other ways to trigger regeneration. After research, data was collected, organized, analyzed, and presented. In my research, I will touch base on a few concepts, such as collagen and cell division. Collagen is a protein fiber that covers the wound, which forms scar tissue during the wound healing when someone is injured. The several categories of collagen all have different functions. Type 1 and type 3 collagen creates scar tissue, while bones are formed by type 10 collagen. Mitosis is cell division. Humans can replicate billions of cells per day using mitosis. However, salamanders use this process to regrow a limb. Scientists have proven that the evolution in humans may have suppressed this rapid or uncontrolled cell division to prevent cancer at the expense of regenerating tissue and regrowing a limb. A wound epidermis is formed when a salamander's leg is cut off. The wound epidermis creates a wave of chemical instructions. Nerves start to stream back into the wound, creating a fiber called the blastema. The blastema is the bud of a new limb of undifferentiated cells, which is capable of growth, or in this case, regeneration. Stored within the blastema are cells known as pluripotent cells. These cells take up their original position and form new muscle, cartilage, and bone. This process is known as mitosis. Before regeneration can undergo, we need to get rid of all the dead cells and pathogen at the wound site. This is the job of the macrophages. They are used to repair muscle. Doctor James Godwin of the Australian Regenerative Medicine Institute decided to decrease the amount of macrophages in a salamander. When removing the macrophages causes regeneration to delay. When all the macrophages were removed, it formed collagen. Scientists have proven that when a tadpole loses its tail during the embryonic stage of development, it is able to regenerate it back. Scientists have proved this with mice, which are also mammals. Yet to be proven with humans, scientists believe vertebrate embryos can regenerate the damaged limb. Gene editing is when you replace a specific section of a gene. Gene editing is banned by guidelines, laws, and regulations in most countries because gene editing on healthy embryos brings up the possibility of irreversible mutations. There are two factors to take into consideration when talking about genome editing: the molecular scissors, which is Cas9, and your guide molecule, which is your RNA. The guide molecule can also be referred to as the bloodhound. The bloodhound leads Cas9 all around the gene site until it finds the specific gene. Then Cas9 cuts off the gene disabling the gene, and is then replaced by the new DNA sequence from your RNA. Ribosomes then build up this new DNA sequence with the proteins required. I also did some more research on the salamander species known as the axolotls, the basic building blocks of life known as the cells, and one of their components known as the nucleus. DNA from the nucleus is condensed into units called chromosomes. There are 46 chromosomes are located in human body nuclei. Chromosomes must be duplicated in interphase before mitosis can proceed. We count the chromosomes by the amount of centromeres present. Chromatids are strands of replicated chromosomes. Even with two chromatids, there is always one centromere. For every the centromere is the part where sister chromatids are held together. For every centromere, there is one chromosome and two chromatids, making a chromosome. I will now explain this. Chromatids are like beans, while the centromere is like the glue that holds them together. that glue attaches two beans together. All with the two beans in the glue, that all forms the chromosomes. So with that one blob of glue, there are two beans. And there are two beads, and that's what it means when you say for every 1 centimeter there's two chromatids. But for every 1 centimeter there's also one end product, which is your chromosome. Now, mitosis is when the adult cell separates to two daughter cells. For the lizards, this process allows them to recover from a lost limb. Specific jumping genes need to be shackled or they will interfere with the process. After replication, 46 chromosomes are created and 92 chromatids. There are four stages of division in mitosis, and these stages are known as PMAT: prophase, metaphase, anaphase, and telophase. Prophase is condensing chromosomes. The nucleus is still present. Metaphase: the nucleus has been disassembled. Chromosomes have lined up. In the anaphase, the chromosomes split into two sister chromatids. Each chromatid is moved away from the center toward the poles or the sides of a cell. Chromatids are moved by string-like fibers known as spindles. These spindles these spindles pull the chromosomes to the centrioles. These spindles are also attached to the centrioles. The centrioles are located at the sides of the pole. The last phase is telophase. Chromosomes are detached from the string-like fiber. Both pairs of 46 chromosomes are at the opposite side of the poles. The nuclei start to form around each pair of chromosomes. So now we have two nuclei in one cell. But cytokinesis then splits the cytoplasm, which is kind of like the between the in between of the two nuclei. And then cytokinesis completes after PMAP stages are done. With all this information, if regeneration has been found in specific organisms as a result of mitosis, such as salamanders, then mammals, including humans, may regrow limbs following controlled mitosis. My data analysis for this regeneration shows the results for various species. My first three species are mammals: the human, mice, and the macaque monkey. There was no regeneration and no limb was replicated. It took about 4 weeks to form collagen. But in the case of their embryos, they were actually able to regenerate both somatic and viscera perfectly in a much shorter time. My snakes, rattlesnakes, python, and boa constrictors all regenerated partially because there was no spine or bone. This took 6 days with regressing. Regressing is the process of referring to its original state. Axolotls, axolotls and leopard gecko are my last two species on the list. There are two types of axolotls: aquatic and terrestrial. Terrestrial axolotls are also known as morphed axolotls. All three regenerated the somatic and viscera. It took the leopard gecko 30 days, the aquatic axolotl 40 to 50, and the terrestrial axolotl 155 to 180. I hypothesized that limb regeneration can be achieved through for research study and testing. Both salamanders and humans use mitosis, but salamanders can use this process to regenerate a limb. Humans may have actually been had the ability to regenerate too, but it may have been suppressed in humans due to evolution to prevent cancer at the expense of regrowing a limb. Another possibility was genome editing. Although this process is banned and illegal globally, except under specific guidelines, this step is a potential field of research study to help with limb regeneration in the future. The time it took for snakes to regenerate was unexpected, as it only took 6 days. Here are a few reasons for that. Snakes can only restore the part of a tail that does not include any organs, so the last quarter to the last third of their body. This is a very tiny section, so it will take less time. Do not forget they only regenerate the somatic, so no viscera, shortening up the process. This article seemed to be cutting about chunks cuts, not the tail amputation. The reason for putting color under the quality of regeneration for the leopard, gecko, and axolotl is because axolotls have plug-and-play body parts, meaning that they can grow a different colored limb, while leopard geckos never have the same tail or limb as the old one. Finally, there were no reptile embryos included in the data table. The reason for this is that most reptiles can rejuvenate a section of their body during and after the embryonic stage, so I didn't see the need to record it. In conclusion, this analysis focused on limb regrowth in various organisms and the challenges to achieving this in mammals. Findings from data collection have shown that certain mammals can regenerate their stem cells during the embryonic stage of development. Hence, if we could study how we replicate embryo stem cells, we might find a way to regenerate a limb. However, I do not believe that we can use mitosis to restore limbs in humans. One successful way will be to use genome editing. For example, only salamanders can regenerate entirely before and after the embryonic stage of development. Scientists believe that something in their genes is unique from humans and other animals, allowing them capable of this phenomenon. More so, the possibility that the mitosis may have altered and suppressed in humans to prevent cancerous growth may have resulted in the inability of humans to regrow a limb. Here's my citations 1, 2, and 3. I would like to acknowledge Weber Academy for this enabling environment and opportunity to perform this research, especially Mr. Bailey and Ms. Summerscales. I owe a great deal of gratitude to Mr. Bailey and Ms. Summerscales for the opportunity to participate in CYSF. In addition, I want to thank my family members for their moral support and contributions. Thank you for listening to my presentation. Hope you enjoyed.